So what I'm going to, as Dr. Vinay said, that we have a very heterogeneous audience and these sessions are meant for everybody. So what I'm going to discuss today is what are the various approaches which scientists used to make vaccines against COVID-19. And as you know, last year, our lives have changed because of SARS-CoV-2. And what we are looking forward in 2021 is to come back to normalcy as early as possible. And for that, probably the only thing is preventive vaccine. Before we go to the vaccine, we all need to know how an immune response is generated to this virus, because the vaccines utilize our own immune system to generate immune response. So SARS-CoV-2 is an RNA virus, and on its surface, it has its major immunogenic protein, which is the spike protein. Once the virus enters our body, it gets into the antigen-presenting cells, where it is broken down into pieces, and the antigen is presented in context with our MHC to the T cell. Once our helper T cells, what we call as CD4 T cells, see this antigen, they get activated and they provide help to B cells. The B cells are the ones which differentiate into plasma cell and produce antibodies. We are all aware that antibodies are protective to some extent, and the major mechanism by which antibodies cause protection is by neutralizing the virus or causing killing of the virus. In addition, the helper T cells also provide help to the CD8 cells. This is especially important in viral infections, where these cytotoxic T cells cause killing of the virus-infected cells. And as we have for other vaccines, we also want memory, that our body should know that we have been immunized against this virus, and if any one of us encounters the virus, we should have protection. And that is what we call as immunological memory, which should be for both B and T cells. So if you see here, the SARS-CoV-2 virus, initially it is, as is true for every viral infections or any infection, initially you get innate immune response, and then we get an adaptive immune response. And as I showed you, that you have T helper cells, you have cytotoxic T cells, and you have a special type of T cells which provide help to B cells, and we generate a memory cells. So this is showing you a speculative dynamics which has also been proven to certain time point, that you have an antigen, the virus, which is shown in purple, and you generate specific T cells. And we hope that these T cells persist for years. And that has been shown for many other viral infections. Similarly, you develop antibodies, the antibodies wane off over time. And people have shown that in COVID-19 infection, there is waning of antibodies after 6 months. But we still have the memory B cells which persist for years. Vaccine development is a multi-step process, and it's not easy to develop vaccines. In the past, it used to take 10 to 20 years to get an effective vaccine. And the three most important steps are the preclinical testing of vaccines, where the scientists try to find out the best antigen possible, which will elicit a very robust immune response. And to further augment the response, we also use adjuvants, which activate the immune system and cause more activation and better immune response. So once we get the best antigen source and the best adjuvant, then they are tried in animal models. You generally start with small animal model like mouse, followed by ferret, which is a model for SARS-CoV-2, and then non-human primates. Once the vaccine has been proven efficacious in animals, it's tested in humans. The phase 1 trials are predominantly to look at toxicity. And then you have phase 2 trials where about 100 to 1,000 people are vaccinated and immunogenicity is seen. And the proof of the pudding is in showing the efficacy, which is done in the phase 3 trials. And as all of you are aware, that phase 3 trials in SARS-CoV-2 infection-related vaccines have been done in 25,000 to 50,000 individuals. Once the vaccine proves efficacious, then only it comes to the market and it is used for mass vaccination. The vigilance doesn't stop there. You still need to monitor people for rare side effects. So that is why it used to take decades to get a good vaccine. But in this era, with better knowledge of science and information from previous platforms which have been used for vaccine, we have been able to compress this period in a matter of less than a year. So this was an editorial in NEJM in May 21, when it was thought that can we develop COVID-19 vaccine at the pandemic speed? The way the pandemic was spreading, would we be able to get vaccine at that speed? And then you have this project, Lightspeed. And you have seen that in January 12th, 2020 was the day when the SARS-CoV-2 genome was sequenced. And in November 18, 2020, we had a vaccine which had 95% efficacy. I mean, this would have been unimaginable before 2020. And all this has happened because of support, both from government, non-government organization, collaboration between countries, and partnership between researchers and the industry. Coming on to the vaccine landscape, it just means that what are the various platforms or antigens which we can use for making vaccines. The nucleic acids, which carry all the message for all the proteins which the virus have. You can use DNA, you can use RNA. These are very easy to make because nowadays to synthesize nucleotides, sequence of nucleotides is very easy, but it has never been done before. Then you can have the live attenuated vaccines, which are the actual virus which have been made weakened so that it generates an immune response, but it does not cause the disease. Or you can make the virus inactivated by using some chemicals or other methods to kill the virus, but it still retains its property of having the antigens on its surface, but it cannot replicate. So inactivated viruses are very safe because they are already dead. Or you can use a small part of the protein, the subunit vaccines, like you're all aware of hepatitis B vaccine, which is a subunit protein vaccine. So these are very, very safe vaccine, but because you're using a small amount of protein, it may be that you don't generate a great immune response. And finally, you can fool the immune system by having an outer coat of a virus which is harmless. You can use measles, you can use rabies, and inside you can put the genetic material of SARS-CoV-2. That is viral vector-based vaccines. So among these, most of these vaccines cannot be used as single-dose vaccines, while the replicating viral vectors or live attenuated vaccines probably can be used as single-dose vaccine. The DNA and RNA-based vaccines have never been used before, and so has been the non-replicating viral vectors. And what we see today is the vaccines which are being used are the RNA-based vaccine and the non-replicating viral-based vector-based vaccine, which we are all getting, that is Covishield. So these viral vector-based vaccines can be produced at a very high scale of production they can have. And similarly for the nucleic acid-based vaccines can be made at a very fast speed. So people have used all these advantages to make vaccines for COVID-19. This is the WHO website, which tells us how many vaccines are in the pipeline. So you have, this is this week's data, that 173 vaccines are in preclinical development and 64 vaccines have moved to clinical development. That means they are in clinical trials. Of these, the majority are protein subunit vaccine. As I told you, they are most safe. And then you have non-replicating viral vectors, and then nucleic acid-based vaccines like DNA and RNA-based vaccines. We all want to have vaccine which is a single-dose vaccine because you know the logistics of giving multiple doses in a large country and to vaccinate the whole world population is not easy. But there are only 20% of vaccines which are being tried as single dose vaccine. Most of the vaccines which are in the pipeline are 2-dose vaccines. And in some, we still don't know that would you need 1 dose or 2 doses. So among these vaccines, there are 19 vaccines which are in phase 1 trial, there are 24 vaccines which are in phase 2 trial, and there are 22 vaccines which are in phase 3 trial. And 10 vaccines have been given authorization for emergency use, and which have been rolled out in many, many countries, and including our own country. So what we said about the immune response, have these vaccines been able to generate of immune response? This is the data of the mRNA vaccine done in non-human primates, which shows that you get good antibody titers with a higher dose. You also get inhibition of binding to the ACE2 receptor. You also get the virus neutralization by this vaccine, the antibodies which develop following the vaccine. You get memory CD4 cells. And finally, you also get that it does not infect the lung tissue. The virus is not able to infect. What I said that we also want a T cell response besides the antibodies. And in this is adenovirus based vaccine. You can see that as the dose increases, low dose, mid dose, and high dose, there is good number of IFN gamma expressing T cells being generated, both at day 14 and day 28. And there is only a minor difference between low dose and mid dose. And the other thing which says how robust is the T cell response is how polyfunctional those T cells are. That is, how many different cytokines do these T cells produce. And if you see here that most of the T cells, CD8 cells and CD4 T cells which have been generated are predominantly polyfunctional, that they produce more than one cytokine. So that shows that we are generating a robust immune response by using these vaccines. And Dr. Gagandeep will cover the clinical efficacy, but this is the RNA vaccine that has shown a great clinical efficacy in preventing infection. And this is our endpoints for vaccines. That one, we want to prevent infection. If we can't do that, can we prevent disease? If we can't prevent infection, can we prevent hospitalization? Can we prevent patients getting admitted to ICU? Or can we reduce mortality? And finally, for the society, can we prevent transmission of disease? We still don't have answers to all of them. The other issues which are there with vaccine are that we are unclear. How long is the duration of protection? Would it prevent an asymptomatic infection? Would it prevent transmission? And finally, that would it cause vaccine-associated enhanced respiratory disease? This happens because when you develop, we want to develop only neutralizing antibodies, which will neutralize the virus. But if you also develop non-neutralizing antibodies, they can activate the macrophages and cause production of pro-inflammatory cytokines, which can cause tissue damage. Or these non-neutralizing antibodies can also cause a increased uptake of the virus and increase in viral load sometimes. So what we have done so far, that we had a long way to go and we have reached quite far. And I think as a scientific community, we need to be proud of that. Till we get the final answer, this is our best vaccine. Thank you.